Hey guys, welcome back. We should start calling this channel Joel Does Biology, right? But anyways, chapter 1.5 is going to be all about viruses and viral particles. This is what I would call like a medium yield. No, it's not low yield, it's not high yield, but you'll get a good amount of questions. So first, let's go over the structure. So you need to know that viruses can be DNA or RNA, and they can be single or double stranded. Okay. So your caspid is low key. It's something like this. So you have this outside, and then you have your DNA, RNA. So it's like a protection. Okay. And then some viruses also have lipid envelopes, and this actually makes them more susceptible to heat and drying out. So, a few things about viruses. They cannot live on their own. Okay, viruses must. They're parasites. I mean, they just can't live without a host cell. They don't have ribosomes, and they can't replicate on their own. So they have to hijack the host machinery to replicate. So, life cycle. So, first step of the life cycle is infection, right? Because we know that they can't produce or live on their own, they have to infect something. So, a bacterial phage, right? So, a bacterial phage is they use fibers to recognize bacteria and a tail sheet to inject DNA. Okay, so once your bacterial phage connects, or let's say attaches is a better word, um, it can fuse in using endocytosis, it can fuse in the membrane. So step two is going to be replication. So viral genomes is replicated and proteins are translated during this stage. DNA viruses use host nuclei. RNA actually often replicate in the cytoplasm. Retroviruses, a good example is going to be HIV, use reverse transcriptase. Now, if you don't know what that is, sorry, we don't have time to go over it, but you should, hint, hint, you should know. So we use reverse transcriptase to make DNA from RNA, and then that's step two of a viral life cycle. And then step three is going to be your proeugenic release. So pretty much the cell lysis. So it's like, let's say that we have a cell, and the cell is at this point, it's like lysed. And then these are like your viruses. Your viruses will be leaving the cell and going off into other shit. Okay? So that is your life cycle of viruses. So next, let's go into some viral genomes. So you have a positive sense RNA, which can be directly translated, and you have a negative sense RNA, which actually requires you have to have a complementary strand first. So retroviruses are definitely a special case. So they actually reverse transcription. So we go from RNA to DNA, then to the host genome. This is actually going to be permanent until the host cell dies. Again, example is HIV. I didn't write anything about bacteriophages. So you have two different cell cycles. In your lytic cell cycle, your virus takes over and bursts, okay? And it does it quickly. Let's put quotation marks quickly. I say that because the main difference is with this other cycle, you actually inject in the host genome and you wait. So your virus doesn't immediately lyse the cell. It can actually lyse the cell, I want to say, whenever it's ready to. And it will eventually do a lytic. So eventually you will get here. But the only difference is with this cycle here, you have to, like, it takes longer, it takes more time. So last but not least, let's go over the subviral particles. So viroids are small circular RNA molecules. They mostly infect plants, but that's actually how we get hepatitis D, because they can sometimes infect humans. I would start that as well. And then they can silence gene expression. Again, I would start that. So prions. Now, we actually don't know. It's actually pretty, I think it's cool. We actually don't know a lot about prions, which is, I find interesting. but they're infected with proteins. They have no nucleic acids, so they're pretty much misfolded proteins, right? So you have an alpha helix that then turns into a beta sheet. So if you, I was about to draw alpha helix, but if you know anything about this mutation, don't worry, I'm making biochem videos soon, so we'll go over it. But this is huge, right? So what happens after that mutation is it leads to aggregation cell death. And a big example that people don't know is mad cow disease was actually a prion disease, which is really interesting, which made it so uncontrollable. Awesome. So that's it for that. Bacteriophages we'll go over soon. I don't know. I didn't put my notes there, but that's it for that, and that's the end of chapter 1.5. Next, we're actually on to chapter 2, which is the cell cycle and mitosis. So I see you there.